Hi everyone, my name is Liz Brower, and here we're going into Module 2, Promoting Host Resistance for Plant Immunity, University of Ottawa in fall 2020. So I just want to give you a very brief overview of a couple concepts in epidemiology. I think most of the content is going to be online for this week, but there's a couple things that I wanted to discuss with you over video, but I'll keep it short. So in plant pathology, one very big area is tracking disease epidemics, as you can imagine. So there's a significant effort to understand the interactions and model them in order to be able to track disease or predict where disease is going to happen next. And so to understand how we do that, there's a couple of things you need to know. In temperate climates like our own, pathogens will produce primary inoculum at the end of the winter and early spring. So that primary inoculum will infect the plant, and usually that's things like spores or bacteria. And then the plant will be infected and produce a secondary inoculum. So that infected plant will then be able to produce this inoculum that will spread to nearby healthy plants, and so on until disease accumulates within a field. So monocyclic pathogens are essentially only producing primary inoculum. So they only have one infection cycle per crop cycle. Polycyclic pathogens have multiple infection cycles per crop cycle, so they will complete this entire loop. In some cases, this can be as fast as 5 days to do this entire cycle. And in the case of, this is the case for Phytophthora infestans, which caused the Irish potato famine, as we discussed last week. So when we're tracking epidemics, one of the things that people commonly do is they just track disease progress curves. So you're looking at how much disease occurs within a field, so disease incidence, or the severity of disease. So, for example, rating how big lesions are on a plant versus the number of plants within a field. So disease can occur either as a linear form over time or exponential growth rates over time. So linear diseases tend to be different types of pathogens from exponential types of pathogens. So we can distinguish between these two different things, and we can apply treatments to try to slow their rate of disease progress. In general, both disease progress curves still have a latent period between which the pathogen has to enter the plant, infect the plant, and produce that inoculum. And then over time, of course, the amount of healthy plants within an area will be saturated, and so you'll eventually reach 100% disease, either incidence or severity. So polycyclic diseases can be much more dramatic than monocyclic diseases, but I just want to suggest here that monocyclic diseases can still be a very big problem. So this is this example is the Dutch elm disease, for example, and over a number of years, we see exponential growth of this pathogen in North America. So this pathogen is actually a oomycete pathogen vectored by beetles. It came to North America from Europe in the 1930s, and the strain of the pathogen that came at that time was much more virulent than the elm trees in North America were able to resist. And so it very effectively killed 40 million trees over a number of years. And this really changed the landscape in North America, because we used to plant a lot of elm trees in neighborhoods because they're quite beautiful trees, or even on farms because they were a very effective windbreaker. But it's very rare to see a very old elm tree now in North America, and that's because of this disease. So part of the problem here was also monoculture, actually. The elms that we were planting were very genetically similar to each other, and so when the disease came in, they're very closely planted as well. It just went through the population in North America. So epidemiology is important. It allows us to track and predict disease outbreaks. And there's a number of different tools that we don't really have time to get into in this course, but I want you to be aware of them. So basically there's groups like this one, and this is the paper, the associated paper if you're interested. But basically what these groups are doing is placing sensors around infected fields, and these are spore traps. So fungal spores that are being airily distributed can be detected by these spore traps, and then these people will take that information and incorporate them into a mathematical model to predict, based on the wind and weather, how this pathogen is going to spread to the neighboring fields. And you can see it's very dramatic. They're obviously able to move quite far distances. And for some pathogens, like fungal pathogens, this can be a very big problem and very hard to control. Things like rust pathogens, for example, can travel thousands of miles. Okay, so this type of modeling and this type of approach actually is used actively by the farming community as well. So it's not just researchers who are doing this. Basically, researchers can come up with a model or an algorithm, for example, that they can sell to farmers, and the farmers can download an app on their phone, and the app will tell them when the environmental conditions were right for a specific type of disease. And then they can apply their pesticides in a timely manner and to try to prevent as much disease as possible. And so there is industry out there who are doing this. And so if you're interested in developing this type of skill set, there is some demand in terms of employers for being able to use this. So Dawncast, for example, was produced by someone at University of Guelph and was sold to this particular company. Okay, so that's all I want to say at the moment, but check out online. There's a lot of information there. And we will be joining you on Friday for a discussion and a journal club. So we'll have our first journal club. Make sure that you read the article and be prepared to talk about your thoughts on that article. Okay, so I'll see you soon.